So hopefully it's going to be better now, and you already saw a glimpse of what I'm going to show you. But as I said, in general, our aim in Melina's lab is to really understand in detail how mammalian eggs develop using advanced microscopy techniques. And in particular, I have done some exciting work that demonstrates what actually happens to those mammalian eggs as the women get older that may impair egg quality. So, as this audience knows best, at fertilization, the egg and the sperm fuse to form a new organism, an embryo. Therefore, quite understandably, having good quality eggs is crucial for producing good quality embryos. And one would therefore expect that the process of egg production is very robust and very efficient. However, this is actually not the case. So when we look at any other cell types in our body, any other mitotic cells, well, the majority of them are fine, and only less than 1% are expected to contain a wrong number of chromosomes. And human spermatocytes aren't very far off. However, when we look at eggs even from young mice, already 4% of them are expected to be chromosomally abnormal, and the incidence of faulty eggs increases as the mouse gets older. But what is actually really surprising is that already 25% of eggs from young women, and by that I mean women in their early 20s, are expected to contain a wrong number of chromosomes, and the incidence of faulty eggs really increases as the woman gets older. But of course you may say, well, we have so many eggs in the female body. So does it actually matter that many of them are not quite right at the level of the DNA? And the answer to that is yes, it unfortunately does matter, and this is because only one egg is ovulated per cycle. And unfortunately, the majority of embryos that come from faulty eggs do not even manage to implant, and the ones that do implant, so they give rise to a clinically recognizable pregnancy, so the woman herself knows that she's expecting. Well, unfortunately, the majority of those still cannot develop fully, which gives rise to spontaneous abortions later on in pregnancy. And in fact, very few full chromosomal abnormalities in humans are viable, the most well-known example being Down syndrome, and the individuals affected by the other two conditions that I'm presenting here, unfortunately, usually die early in childhood. So I hope that you would agree with me that understanding how mammalian eggs develop is very important. So what is actually so special about the process of egg production that may explain this apparent inefficiency? Well, eggs are produced in a specialized cell division process called meiosis, and the general outline of meiosis has been really known for quite some time. So it starts with a premature egg precursor cell called an oocyte, and the nucleus of an oocyte contains 23 pairs of chromosomes, so one set from the mother and one set from the father. And at the other end of this process, so at fertilization, the egg and the sperm fuse together to form a new organism, an embryo. And because both the egg and the sperm provide genetic material to the embryo, for the embryo to contain the same amount of DNA as the parents, the oocyte has to have the amount of its own genetic material. And this is achieved through two subsequent cell divisions called respectively meiosis I and meiosis II. So in meiosis I, the aim is to actually separate the two chromosomes, so the maternal from paternal. So again, we start with 23 pairs of chromosomes and we try to separate them from one another so that you retain only one chromosome of each pair inside the egg. On the other hand, meiosis II really resembles any other mitotic cell division in our body, in the sense that we start with a whole chromosome and we try to separate the two sister chromatids. So again, at fertilization, the egg provides a set of chromatids and the sperm provides a set of chromatids, giving the embryo a full set. But this is actually the same between males and females, so the fact that we have two divisions. And I said to you in the beginning that it's actually female meiosis that is so prone to errors. So what is actually so special about female meiosis that can explain this? Well, female meiosis is really exceptional because of the time scale at which this process occurs. So while in males the spermatocytes are produced within 72 to 74 hours, in females the oocyte precursor cells are already formed during the fetal life of the woman, so when the woman herself is in utero. And on the other hand, the second meiotic division is only completed upon sperm entry at fertilization. So what this essentially means in practical terms is that if a woman were to become pregnant in her late 30s, and as we know many women do, the oocyte that would be fertilized would be involved in this process of meiosis for several decades. And this in fact gives this chromosome segregation machinery a substantial amount of time over which to deteriorate. So what actually happens to the chromosome segregation machinery as the woman gets older? Well, so as we said, in meiosis I, we try to separate the chromosomes, and in meiosis II, we try to separate the chromatids. But before we can do so, the two chromosomes have to be linked together so that ultimately they can separate. And this tight association between the two right chromosomes and between the correct sister chromatids is maintained by what we call the cohesin proteins. So the cohesin proteins here in pink, really, they essentially act as chromosome glue. So they really stitch the right chromosomes and the right chromatids together. And it has been very interestingly demonstrated recently that cohesins actually have a very minimal turnover. And in fact, they can retain their functionality for a limited amount of time. So what then happens is that when the woman gets older, some of those cohesin complexes get lost, and this leads to this loosening up of the chromosome architecture. And this really challenges the chromosome segregation machinery. So this was a very important discovery, and it really provided us with some insights into why meiosis in mammals is so error-prone. However, we still think that it doesn't explain fully this striking increase of aneuploidy with advancing maternal age. And at this point, of course, you can wonder, like, considering how important this question is, why is it actually the case that we still don't have an answer? So why do we still not know fully why the quality of eggs declines with advancing maternal age? And the answer to this question is actually very simple. And this is because studying female meiosis is actually very challenging. So first of all oocytes are primary cells, which means that even if you work with mice, whenever you do an experiment, you actually have to retrieve the oocytes directly from the mouse. And secondly, oocytes are transcriptionally silent, and they have this very stable big pool of proteins, which essentially means that it's very difficult to study gene and protein function in oocytes. And if this hasn't been enough, studying the maternal age effect is actually even more challenging. So first of all, we were able to demonstrate in the past that some aspects of the deteriorating chromosome architecture in human eggs are not very well mimicked in mice. So even though mice are an excellent model for studying meiosis, perhaps we can't use it to study some of those age-related defects. Secondly, apart from mice, there are not really any model systems available to study mammalian meiosis using microscopy techniques. And of course, you can say, like, why don't we actually address these questions directly in human eggs? And that would, of course, make a lot of sense. But unfortunately, the supply of human eggs available to research is quite scarce, so we can't address all the questions directly. And this really translates into not many labs having had the opportunity to work with those human eggs. So there aren't really many well-established tools or protocols available. But because Melina and I really wanted to understand what happens to those eggs as the woman gets older, for the second part of my PhD, we thought that if there are no tools available, I can just try to design those tools. But instead of talking about like the technical aspects of it, I'm just going to tell you now about what we found out from this analysis. So as I said to you in the beginning, with age, those cohesin complexes, so this chromosome glue gets lost, and this leads to this loosening of the global chromosome architecture. But Melina and I thought that, okay, perhaps it's not just the cohesins, but perhaps there are some other aspects of the chromosome segregation machinery that also deteriorate, that have been missed to date. And we thought that perhaps kinetochores are very good candidates for this. So kinetochores are here demonstrated in pink. And essentially, kinetochores are those protein-rich structures that assemble here on the centromeric region, so the middle region of the chromosome arm. And they're very important because, as we said, in meiosis, we try to separate the chromosomes. But in principle, this chromosome segregation machinery, so the spindle microtubules, they don't pull on the chromosomes themselves, but instead they pull on those kinetochores. So kinetochores really act as an interface between the DNA and this chromosome segregation machinery. And we thought that the kinetochores would be actually a very good candidate for this age-related deterioration for two reasons. Firstly, when I say they're protein-rich, what that actually means is that they're composed of 100 different proteins that have to exist in a very precise stoichiometric balance. So if you think about it, if something is that complex, it's very likely that it can go wrong over those decades of arrest. And secondly, kinetochores are very important because they, as I said, they really act as this interface, so they drive chromosome movement. And as we know, aneuploidy aneuploidy comes from chromosome segregation errors. So it would make sense that if something goes wrong with the kinetochore, so this structure in pink, this could really affect chromosome segregation outcomes. And very interestingly, it has been recently demonstrated in 2016 that just like cohesions, at least one protein that really defines kinetochore assembly, called SEMP-A, is actually also long-lived, so it has this very low turnover. And in fact, it declines with age by 30%, at least in mice. So we thought that, okay, perhaps it's not just the cohesion complexes, but maybe kinetochores themselves deteriorate as well. And this is something that we wanted to investigate. So is it actually a missed component that people have missed to date? So do kinetochores deteriorate as well? And to do so, we decided to look directly in Meiosis II. So from now on, we'll focus on Meiosis II only. And this is because in Meiosis II, the chromosome is made of those two chromatids, and the kinetochores are quite well separated, which really allows you to investigate individual kinetochores. So this is an immunofluorescence, so a microscopy image of a chromosome from a young mouse. And here you can see the chromosome, so the DNA being labeled in blue, and the kinetochores being labeled in pink. And one thing that I'd like you to remember throughout the talk is that there's always one-to-one stoichiometry between the chromatids and kinetochores. So in Meiosis I, if you have 4 chromatids, you have 4 kinetochores. In Meiosis II, a chromosome is made of 2 chromatids, so 2 kinetochores, and if you only had 1 chromatid, 1 kinetochore. So it's always like 1-to-1 ratio. So again, this is a chromosome from a young mouse, and because we have 2 chromatids, you expect to see 2 kinetochores, and this is indeed what you see. So you can see those 2 fluorescent foci. And I have to say that was not that interesting. However, when we looked at chromosomes from aged mice, it turned out that they actually look quite different. So I hope that you can really appreciate that here the kinetochores look quite different. So they're, what we say, dissociated. So they're not very nice and compact, but instead they have this stretched dissociated morphology. And this was very unexpected. So we really decided to investigate this in more detail. And in fact, we looked at over 6,000 kinetochores from mice of different ages. And really 2 things emerged from this. So first of all, that around 35% of all kinetochores in aged mice show this dissociated pattern. And secondly, that the degree of dissociation actually increases with advancing maternal age. But of course, these were the mice. So then we wanted to see, is it something that is mouse specific? Or does this kinetochore dissociation also occur in human eggs? So then we had a direct look at human eggs. And here's an example of a chromosome from a young 27-year-old woman. And again, the DNA is labeled in blue and the kinetochores in pink. And I hope that you can really appreciate that even though these are 2 kinetochores of the same chromosome, so you'd expect them to look really exactly the same, they actually look very different. So the left and the right are not very symmetrical. So while this kinetochore is nice and compact, this one really shows this dissociated morphology. And this was very interesting because you would think that the 27-year-old woman is a young woman, so you would hope that her kinetochores would be still very nice and compact, but this indeed may not be the case. And in fact, we were able to demonstrate that indeed also, just like in mice, in women, the kinetochore dissociation also increases with age. Okay, so at this stage, we knew that kinetochores are not always compact, and instead, with age, they become more dissociated. But how could that actually affect chromosome segregation outcomes? So we know from mitosis that chromosome segregation errors really arise from abnormal kinetochore microtubule interactions. So in green, these are the spindle microtubules, so this chromosome segregation machinery that pulls on the chromosomes. And in Meiosis II, we really try to separate the 2 chromatids, so to pull them apart. And chromosome segregation errors arise when one of those kinetochores is actually attached to those microtubule bundles that come from different spindle poles. So if you then imagine what happens at anaphase, as I said, the aim is to pull them in opposite directions. Here, this one kinetochore would be attached from both sides. So it would be pulled from both directions. And what that essentially means, it wouldn't really know where to go. So it would go a bit to the left, a bit to the right, but essentially it would just stay behind with what we call lagging. And it has been demonstrated very clearly that those lagging chromosomes are the ones that cause problems. So these are the ones that missegregate. So considering that now our kinetochores look quite different in aged females, so they had this dissociated morphology, they were composed of those two lobes. We wondered whether those dissociated kinetochores are actually more likely to display those abnormal attachments. And this indeed turned out to be the case. So the abnormal attachments are what we call merothelic attachments, and you can see that the dissociated kinetochores are more likely to display those merothelic abnormal attachments. So just to summarize at this stage, we're able to demonstrate that kinetochores are not exactly, they don't really look the way we always imagine, so they're not always nice and compact, but instead they change morphology with age, and predominantly they dissociate into those two domains. And also that in humans, even what we think are young women, at least from their reproductive perspective, they already have quite many of those dissociated kinetochores. And further, that the kinetochore dissociation actually affects the way in which kinetochores interact with those spindle microtubules, in the sense that they display most of those incorrect attachments. So this was a very interesting observation, especially for people who, like myself, who are interested in kinetochores. But of course, as molecular biologists, we don't want just to see things, but we also want to understand why they happen. So we really wanted to understand what is actually the mechanism behind this dissociation. Because if you think about it in the future, if you want to counteract it in any way, you first have to understand why it happens, so you can actually design some potential treatments to overcome this problem. So again, this is what we already know. This is what I told you so far. So with age, you lose those cohesion proteins, and this leads to this loosening up of the chromosome architecture. But let us go one step further. So if you get even older, you lose more of those cohesion proteins. So what this essentially translates into is that at some point, there isn't just enough of this chromosome glue to keep the two chromatids together, and they completely separate. And this really leads to problems of those single chromatids because they cannot segregate properly. So if you think about the young egg, it has those very beautiful intact chromosomes only, whereas in an egg from an older mouse or an older woman, you expect to see a mixture of all. So some chromosomes would be still okay, whereas the other ones would be loosened up and some would be completely separated. And looking at those single separated chromatids really gave us a very important first insight into the mechanism. So this is a microscopy image of how people in the field pictured a single chromatid to date. So again, we're looking at a single chromatid. It's always one-to-one, so you expect to see one kinetochore only, so one fluorescent spot. And this is indeed what you can see here. But it turned out that the majority of single chromatids actually look very different. So instead of seeing just one spot, I hope that you can really appreciate that you can very clearly see those two distinct foci here. And this was, again, very unexpected because it's a single chromatid. You wouldn't expect to see anything else than that very single kinetochore. And indeed, it turned out that single chromatids are significantly more likely to show this kinetochore dissociation than their intact counterparts. So both in aged mice and in women, while 25% of kinetochores on intact chromosomes are still like nice and compact. On single chromatids, even 75% of kinetochores show this dissociation. So what this really suggested is that perhaps as the woman gets older and those cohesion complexes, so this chromosome glue gets lost, it's not only that you get this disintegration of the global chromosome architecture, so the chromosomes loosening up and separating into single chromatids, but perhaps that this also impacts on kinetochores themselves. So once cohesion is lost, the kinetochores dissociate. But of course, this is just a correlation. And we wanted to see if this is actually a causal relationship. So is it the case that cohesions have this additional function to protect kinetochore integrity? So in order to do that, we decided to go back to young mice, the kinetochores of which should be normally very nice and compact. And we thought that if we deplete cohesion complexes, so if we acutely get rid of this chromosome glue, would it be the case that then the kinetochores dissociate into those two domains? But I said to you in the beginning that actually depleting proteins in oocyte is very difficult because they have this very large pool of those very stable proteins. But very fortunately, very recently we have developed this new tool in the lab that allows us to acutely get rid of the proteins. And without going into too much detail, this tool that we call TrimAway, it really relies on TRIM21, which is an E3 ubiquitin ligase, but this doesn't matter for now. But what you do is you introduce into your cell this protein, and you also introduce an antibody against your target protein. So then your antibody binds to your target protein, and this protein TRIM21 TRIM21 can recognize this protein antibody complex. And then this actually marks this whole complex for degradation. So what this essentially means is that if you introduce this TRIM21 and the antibody into your cell, you can actually get rid of your protein within 5 to 10 minutes, which is really excellent. So our experimental workflow is quite similar to what you do when you do your ICSI procedure, in the sense that we take an oocyte, we introduce this TRIM21 protein, and also fluorescent labels so that we can actually see the DNA and the spindle microtubules and the kinetochores, so we actually know what's going on inside that cell. And we allow the egg to progress through meiosis so that it reaches the M2 stage. And at this point, we introduce an antibody against our protein of interest. So for me, that would be this cohesion complex, the chromosome glue, and we see what happens. So what this should really allow you to do is to have a very nice young egg, but without this chromosome glue. So a situation when the chromosomes have completely fallen apart into single chromatids. And then you can have a look at this egg and see if in this instance those kinetochores have dissociated. So this is how it looks really in reality. So this is a young oocyte, and you can see the chromosomes being labeled in red, the microtubules, so the chromosome segregation machinery in blue, and the kinetochores in magenta, and it's going to play again. So you can see that within like a couple of minutes, those chromosomes completely fall apart into single chromatids. And then you can take the same egg, you can fix it and do immunofluorescence microscopy and really look at those kinetochores in a lot of detail. So the question then was, okay, is it really the case that when you get rid of chromosome glue, those kinetochores in young women would dissociate into those domains? And this indeed turned out to be the case. So if this is a control chromosome from a young mouse, again, it's a chromosome, so 2 chromatids, you expect to see 2 fluorescent foci. When you got rid of the cohesion glue, in fact, those single chromatids started having those dissociated kinetochores, so you can clearly see those 2 domains. And we tried it for many different cohesion components, and it turned out to be really the case. So that once you get rid of cohesions, the kinetochore predominantly dissociate into those 2 domains. So at this point, you could say, okay, in that case, cohesion loss is sufficient to cause this kinetochore dissociation. But one thing to remember is that we're not really just looking at the chromosomes, but the chromosomes are really in the context of a cell. And they don't just sit around in the cytoplasm to wait for us to do those assays. But the reason why those chromosomes are interacting with the cytoplasmic factors in the first place is because they're trying to become segregated. So they're constantly being pulled by this chromosome segregation machinery, so the spindle microtubules. And it really exerts those pulling forces on those kinetochores. So we started wondering if the kinetochores are being pulled on constantly by those spindle microtubules. Does actually contribute to kinetochore dissociation as well. So if you think about how to address this question, well, you can really do 2 things. So get rid of this cohesion glue and either reduce the pulling on kinetochores or actually enhance the pulling by promoting more dynamics of the spindle itself. And this is essentially what we've done. So we did a similar kind of assay, but using a drug called nocodazole, like low doses of nocodazole, which depolymerizes some of the microtubules, and then we got rid of the chromosome glue. And indeed, when you look at the bars in black, you can see that here the degree of kinetochore dissociation went down. So when you have less pulling, you have less of this disintegration of the kinetochore. And then we've done the opposite assay, which is a bit more tricky, but if you then treat the oocyte with a drug called Taxol, this stabilizes microtubules and gives you this very beautiful enlarged spindle. But then when you wash the drug out, the spindle tries to reorganize back to its nice bipolar shape. But this is what actually then happens to the chromosomes. So they're really becoming, when the spindle tries to go from here to there, the chromosomes are really pulled from different directions. So this really enhances the pulling on them. And then you can again take this egg and fix it and look at the kinetochores. And again, it turned out that under these conditions, so when you enhance the microtubule pulling, the kinetochore dissociation goes up. So what this really essentially means is that cohesion loss and this microtubule pulling really act together to dissociate the kinetochores. And if you get rid just of the cohesions in a young egg, so when you get rid of the chromosome glue, that's already sufficient to cause kinetochore dissociation. So I hope that I managed to convince you that cohesion loss is important, but there are also other factors of the chromosome segregation machinery that deteriorate with age, and that when cohesions are lost, this leads to this loosening of chromosome kinetochore morphology. And we think that this really acts as a positive feedback loop in a sense, so that when the kinetochores become more dissociated, they're more likely to interact with many spindle microtubules that come from different directions, and then this dissociates the kinetochore morphology even further. So what I mean is that if it goes bad, it'll just get worse and worse when you get more and more pooling. So what does it actually mean in practical terms? Well, we think that if you're still young, or at least when you're a young mouse, your kinetochores would be very nice and compact, and they would interact with microtubules that come from one direction only. Whereas the older you get, the more dissociated your kinetochore become, and they become pulled from different directions. And then those chromatids will really cause errors at anaphase because they will probably lag behind and missegregate. And this is really what sets the boundary between like the healthy eggs, the one that you want to be fertilizing, and the aneuploid eggs, so the ones that we would all rather avoid. So at this stage, I would like to thank you all for your attention. Of course, thank Melina, my supervisor, for having given me this amazing opportunity, and especially our collaborators at the Bornhall Clinic in Cambridge and then the Kinderwunschzentrum in Göttingen in Germany for really allowing us to work on those human eggs, and of course the entire SHU lab for their excellent support. Thank you.